Welcome to section 4.3, citric acid cycle and oxidative phosphorylation. In this section, we'll explore the fate of the pyruvate molecules produced during glycolysis and how cells extract even more energy by processing those molecules in the mitochondria. This is where the majority of our ATP is generated. By the end of this lesson, you should be able to describe the citric acid cycle and its products, explain the role of the electron transport chain in oxidative phosphorylation, and understand how ATP is generated through chemiosmosis. These processes are critical for how your cells and all aerobic life power themselves. The citric acid cycle and oxidative phosphorylation are steps in aerobic respiration that take place in the mitochondria. After glycolysis, pyruvate is transported into the mitochondria and converted into acetyl coenzyme A, or acetyl-CoA, which enters the citric acid cycle. The cycle produces high-energy molecules, NADH and FADH2, that carry electrons to the electron transport chain. Let's zoom into the mitochondrion, the cell's powerhouse. This organelle has a smooth outer membrane and a folded inner membrane called a cristae. The citric acid cycle occurs in the mitochondrial matrix, while the electron transport chain is embedded in the inner membrane. This setup is crucial for separating charges and building the proton gradient needed for ATP production. In eukaryotic cells, the pyruvate made at the end of glycolysis moves into the mitochondria, the cell's main site for making energy. If oxygen is present, aerobic respiration continues. Inside the mitochondria, each 3-carbon pyruvate loses 1 carbon as carbon dioxide. The remaining 2-carbon unit, called an acetyl group, attaches to a helper molecule called coenzyme A, made from vitamin B5. This forms acetyl-CoA, which carries the acetyl group into the next stage of energy production, the citric acid cycle. The citric acid cycle happens in the mitochondrial matrix. It's a closed loop, meaning the molecule that starts the cycle is regenerated at the end. Each turn of the cycle processes 1 acetyl group, releasing 2 molecules of carbon dioxide, producing 1 ATP, and creating high-energy electron carriers, 3 NADH and 1 FADH2. These carriers will pass their electrons to the final stage of aerobic respiration, where most ATP is made. It takes 2 turns of the citric acid cycle to completely process the pyruvate made from 1 glucose molecule. Over those 2 turns, all 6 carbons from glucose are eventually released as carbon dioxide. The cycle is not just about breaking things down for energy. It also provides building blocks for making certain amino acids, so it's both energy producing and useful for building cell materials. From 1 glucose molecule, the citric acid cycle produces 6 NADH, 2 FADH2, and 2 ATP molecules, along with 4 molecules of CO2. These high-energy electron carriers are the main output. They'll now move on to the electron transport chain where the bulk of ATP is generated. Most of the ATP made during the breakdown of glucose in aerobic respiration doesn't come directly from glycolysis or the citric acid cycle. Instead, it's produced in a final stage called oxidative phosphorylation. This process starts when high-energy electrons from earlier steps are passed through a chain of protein complexes. In eukaryotes, these complexes are built into the inner membrane of the mitochondria. In prokaryotes, they're found in the cell membrane. The electron transport chain is a series of protein complexes located in the inner mitochondrial matrix. NADH and FADH2 donate electrons to the chain, which pass from one complex to another. As the electrons move through the chain, their energy is used to pump protons across the membrane, creating an electrochemical gradient, a kind of stored energy. The cell then uses this stored energy to power an enzyme that makes ATP. Oxygen acts as the final electron acceptor, allowing the process to keep going. This proton gradient drives chemiosmosis. Protons flow back into the mitochondrial matrix through ATP synthase, a molecular turbine that uses the flow of protons to generate ATP from ADP and inorganic phosphate. This process of ATP generation using the electron transport chain and chemiosmosis is called oxidative phosphorylation. Oxygen is the final electron acceptor in the chain. It combines with electrons and protons to form water. Without oxygen, the entire system backs up and oxidative phosphorylation halts. This is why aerobic organisms require oxygen to survive. Adding it all up, aerobic respiration from one glucose molecule produces about 30 to 32 ATP. Two come from glycolysis, two from the citric acid cycle, and somewhere between 26 and 28 from oxidative phosphorylation. This high yield is why oxygen is so important for energy demanding organisms like humans. The citric acid cycle and oxidative phosphorylation work together to extract the majority of energy from glucose. The cycle generates electron carriers, which feed into the electron transport chain. That chain powers ATP production via chemiosmosis. It's a beautiful example of cellular engineering, one that's essential to almost all life on Earth.